The next slide. ये यहाँ से click करने का next slide नहीं करेगा. Okay, thank you. Okay. So I would like to first thank the organizers for their kind invitation and also the opportunity to share some of our latest work on neurodegenerative diseases. So I will be switching gears towards specifically Parkinson's disease and using the multi-omics approach. So what we are doing in the lab is mainly working with multi-omics data sets. So we generate, integrate, and analyze data sets from different layers. So starting with genome, as you have already heard about the genetic variations, and also then we have epigenome, where we can see that how the chromatin is open and closed, using, for example, ATAC-seq data, and also the transcriptomics, bulk RNA-seq as well as RNA-seq, and then ultimately checking if these all effects has some changes in the protein level. So on top of that, what we also try to do is integrate the environmental exposures, for example, the lifestyle, or what we are, for example, what we are eating, or if we are smoking or drinking, because these environmental exposures can also affect our epigenome, which will ultimately have effect on our transcriptome and proteome. So what we try to do is, using machine learning approaches, we try to learn the patterns which can classify our disease cohort, to say, with the as respect to the healthy cohort. And we can also go further, because this is a binary classification. We can also, within the disease, as we are going towards precision medicine, we can also try to predict the clinical outcomes using this multi-omics data. So what we are using as a model are iPSCs. So we are part of this founding PD consortium, where we have access to almost around 100 iPSC lines. So I will show you some of the data set in some next slides. And then parallelly, we also work with brain tissues to validate if what we are finding in the model can be replicated in the postmortem brain tissues. So we are mainly working with Parkinson's disease since last 3 or 4 years, as this is the second most common neurodegenerative disease, and also the hallmark is the loss of a specific type of neurons that are dopaminergic neurons in the substantia nigra region of our brain. And the 4 motor symptoms, what we have known so far in Parkinson's disease, is the slowness of the movement, the rigidity, and also the tremors, and also when we walk, it's like balance abnormalities. So just to give you some numbers, we have more than 10 million people, but it's predicted to go beyond double or even triple in next 25 years because of the aging population. And if we talk about the financial burden, in US alone, it was calculated over $50 billion per year spending on PD, taking care as well as on the research. So one thing to note here is that we also have in PD not only motor symptoms but also non-motor symptoms, specifically the mental or behavior issues, cognitive issues, as well as the some of the depression symptoms we have. So genetically, we know some of the genes which can be directly related to the Parkinson's disease. For example, one of the first gene, the C-nuclein, where we know that this is causal for PD. But then we also have some of the strong risk factor, GBA1 and LRRK2. So I will be calling it as LRRK2 gene. And then some of the risk factors has been identified by GWAS. So it was also introduced in the earlier talks that we can identify using this cohort analysis, as you have seen in the Manhattan plot, that some of these SNPs can be identified in the population, which can be related to the disease. So one of the things what you will notice here is that mainly these genes has been studied in the substantia nigra region of the Parkinson's disease. So what we are missing is the other parts of the brain which can be associated with mainly the non-motor symptoms. And this is very important because non-motor symptoms can arise in PD way before once you have the motor symptoms. So once we know what are the mechanisms that are triggering the non-motor symptoms, we can start predicting that which person can have Parkinson's disease in the future. So this is exactly we did with our pilot study, which is now published in 2 months ago in Molecular Brain. So we have a small cohort of available post-mortem brain tissues from different regions of brain, and also the mutations which are relevant for Parkinson's disease, and we compare it with the healthy control. So I will just take a couple of minutes to go through the experimental design and also the analysis as we are doing the single nuclei RNA-seq, and we first of all we try to integrate the cell types with the Jiva studies to identify which Jiva SNPs which are related to PD are overrepresented in a specific cell population. So this is what we try to do with the, for example, let's say the Magma approach, and then in Germany we have a separate cohort of PD patients with the clinical outcome numbers around 1,000 patients, and we construct in this independent cohort the polygenic risk score. So coming from genetic risk plus transcriptomics, we try to predict if using the polygenic risk scores we can identify the clinical outcomes, for example the UPDRS, which is a motor symptom, and then we have MoCA and VDI, which are cognitive and the depression symptoms. And then we also validate our findings in the publicly available data sets, specifically in substantia nigra region. So just to start with, here what we have is around 90,000 high quality single nuclei. So each dot here represents a particular, let's say, cell or a nuclei. And we have a cell types, for example, neurons, ODCs, which are oligodendrocytes, OPCs, which are our oligoprogenitor cells, and microglia and astrocytes. So these are all expected cell types which which we normally find in the post-mortem brain tissues. So when we integrate this data set with the PD Jivas, what we find, like what was very surprising, is that normally this is disease related to the neurons, and we find the overrepresentation in OPCs and ODCs. So this is something was very exciting for us as well as surprising for us. So what we did is we have these genes which are misregulated in ODCs and OPCs, and as a control we also use microglia. We try to predict the clinical outcomes in an independent cohort. So here we have motor symptoms and non-motor symptoms. So what we saw here is that using the OPCs and ODC scores, we are mainly predicting the non-motor symptoms, and none of these scores predict the motor symptoms. And specifically also the microglia, we see no prediction. So this is the way kind of what we can try to achieve is that the misregulated genes, they are predicting the non-motor symptoms using a particular risk factors GBA1 and LAR2. So just to check if this is only in the non-substantia nigra region, or we can also predict this some of the motor symptoms in substantia nigra, we use this publicly available data set, which again confirm that going towards the substantia nigra, these OPCs and ODCs can indeed predict the motor symptoms as well. So here we also have not only the cell type specificity, but also the region specificity. So of course, what we wanted to now study was that if these oligodendrocyte cells are like really new players in Parkinson's disease, and we wanted to model this with the iPSC cells. So what we are starting with is already with the these PD lines and controls, and differentiate them to oligodendrocyte lineage cells, and we produce the single cell data, bulk RNA-seq, as well as the cell painting, which is more towards the morphological profile. So this is something we do with the in collaboration with Pullman Lab in Germany. So far, we have generated the single cell data from this particular cohort. And what you can see here is, again, we have the population of oligolineage cells, which we define it using the markers, for example, CMP, PLP1 for ODCs, and for OPCs, we have mainly PDGFRA. And also some of these markers in IPPC population, which are proliferating cells, we can see the multiple markers. So what we have done is like here the subclustering of this population, and we call it as PPC2, 1, and 0. So to confirm this, as we are modeling this in the Petri dish, that is it related to, or can we see similarity between the markers in the post-mortem brain and what we see here? Indeed, when we do the clustering with the post-mortem single nuclei RNA-seq data, we see ODCs are clustering very nicely with the post-mortem ODCs, and same for the OPCs. So just to confirm that the cell types what we are generating is indeed similar to what we find in the brain. And again, what we did is like for PDGFRA, where we have very restricted expression in OPCs for this particular LARC2, which is a PD-related gene, we see more spread of this expression. So what you can see here is in OPCs and epithelial cells, and these particular population of or cell types are overrepresented in LARC2 mutation versus healthy control. So suggesting that there is a direct link, or maybe there is a direct link with the LARC2 expression and the cell composition analysis, what we are doing in the iPSC-derived oligonuclear cells. So next we repeated our analysis with the integration of PD-Jiva. So what we can see here is again a very nice replication what we did in the postmortem brains, that ODCs and OPCs mainly, they are overrepresented in PD-Jiva's risk factors, and this is indeed suggesting us what we are modeling here is capturing our PD risk types. So what we are now trying to study is the mechanisms which are responsible for, let's say, the patterns what we are finding in these PD cell types, and we are doing different kind of analysis, for example, differential expressions, pseudotime analysis, as well as some of the cell-to-cell communication. So just to show you an example, what we have done is taking all these cell types separated by healthy control and LARC2, you can create a network between cell-to-cell chain. So to our surprise, what we see here is in particular IOPCs, we see a reduced connectivity in LARC2 mutated lines, suggesting that some of these pathways are going really down in particular LARC2 mutated lines. So we validated this, that which genes can be responsible for this. For example, in OPCs, we find SEMA6A and PLXNA2 specifically downregulated in OPCs, and we confirmed this also in brain, meaning that this is not only what we find only in iPSC-derived cell types, but also in postmortem brains. So what we are trying to do now is that we have these two mutated lines LARC2. What with the other iPSC lines which have GBA1 or C nuclear mutation? So for this, what we are doing is taking the iPSC lines from founding PD consortium. So it's a big consortium funded by Michael J. Fox Foundation, where we have access to almost 100 iPSC lines, and we have already generated iPSC-derived neurons data from using these GBA1, LARC2, and C nuclear mutated lines, and created the multi-omics data. So now the idea is we would like to also go for the oligodendrocyte lineage cells to see if we can have a specific patterns of this SEMA6A pathway in LARC2 lines, or this is also common between GBA1 or sporadic cell lines. And parallelly, what we are now looking for collaboration is to collect these postmortem brains with different mutations and different regions and different stages of Parkinson's disease, to generate multi-omics data so that we can replicate our findings from the iPSC-derived lines. And indeed, we can see that these changes are on early stages, or these are coming after the diagnosis has been done. So finally, I would like to acknowledge my lab. So we have Mo and Nasser, who did like all the single cell analysis. Also, Nasser did some of the wet lab iPSC differentiation, and the polygenic risk scores is mainly done by Mo and Benhua, and the bulk RNA-seq analysis will be done by Natalia, and then we are also doing some tool development for single cell analysis. For example, Dennis generated this pipeline SC-ROC to refine the cell clustering, and also to check the QC of data. Benjamin did this tool SC-MOMS QC, and also I would like to thank all the collaborators from different institutes, and especially the funding agencies, DFG and Michael J. Fox Foundation. And thank you very much for your attention, and I'm happy to answer your questions.
Deep science and good team. You as a multiomics, the expectation of translation, how far we are?
It's a very good question. We are, I would say, very far away.
Very far.
Yeah. So one of the thing what we are trying. So I'm glad that you asked this question because in the morning, Dr. Goswami also mentioned about the applications, right? So what we are trying to do is once we have these iPSC lines, for example, just imagine that if you have certain mutation, if we generate iPSC lines from your, let's say, some somatic cells, we can differentiate them to neurons and oligodendrocytes, for example, and we can start predicting if you will have a particular disease in future or not. So this is the application what I would really like to pursue in future. Yes, so it's also related to prevention, but also kind of early diagnosis that one can predict if what is the probability of getting a disease. Yes.
Okay. One issue, one particular problem, suppose few patients will see constipation.
Yes.
If we could solve that, lot of improvement.
I would say that is one of the earlier biomarker what we have in neurodegenerative diseases. Yes.
You see, there must be some, the whole understanding is now performing science. People don't want to wait for another 20 years with your science to come out with multiple drugs.
Yeah.
This is what the expectation. So there are alternatives. Of course, we have apple, but what we see is that microbiome.
Yes.
C is playing a very important role.
I agree.
Only simply dysbiosis. We tackle dysbiosis, we are not good with it.
I agree.
So,
I agree. This reminds me what my grandmother used to say: "What you eat is directly connected to your brain thoughts and psychiatry." Yeah.
So I believe the polygen score is based upon accepting the UV based on property.
No, these were in C plus T, so we do the clumping and thresholding. So that is a very simple procedure to do the PRS.
So is it based upon ratio?
No, so this is based on you take the odds ratio and then you calculate what is the like allele 1 and allele 2. Yeah, so basically you are taking the PD jivas and then you take the genetic data from the new cohort and then you calculate based on the odds ratio.
Thank you so much.
Hi, I'm Dr. Sundar from Singapore. Regarding the deep learning thing, right? So from clustering, how you plan to go about classification or modeling or things like that?
Yeah, so this is something ongoing. What we are trying to do here is, so once we have the clustering in single cell data, right? Many times what we see is that the cells what we see in UMAP, they go in another cell type, but they are still annotated as cell type A. And we can clearly see this is B. So it's kind of refining that you can learn the patterns from, let's say, the clusters what you have, and then you run again your model to refine that cluster.
But you should plan for the classification and then we can get the new things, right? Because a lot of things are not unknown or there.
Yes.
So we should get how we are going to put them in a different cluster or how we are planning for it.
Yes.
Modeling.
So that would be the next step. Once we learn the patterns from different cell types, then you start predicting the new cell types even without the information of, let's say, cluster 1 and cluster 2.
Yeah. Okay. Yeah, we can collaborate sometime.
Yes. Sure. Thank you. Thank you. Yes.
So if genetic predisposition is established for cohort and if PRS is threshold is set, then I think genetic predictions can be made by isolating DNA from the blood or saliva beforehand. So what's the extra advantage of making IPC cell lines and then predict the disease?
Yes, that's a brilliant question. So when we take all the SNPs which are associated with, let's say, PD, without information of cell type specific data, we don't see prediction of clinical outcomes. Once you have data set or differentially expressed genes from, let's say, oligodendrocytes, You take only those genes and then you calculate polygenic risk scores based on the SNPs which are nearby those genes. Then you see the prediction of clinical outcomes.
Thank you. Great work.
Thank you.
So with this, we come to the end of the first session with Paul Day. It was very successful because the talk very interesting, which was reflected by the kind of discussion that was generated. And the talks ranged from natural selection to derivation DNA to orchestra of life and then mice, platula, and then finally Bansal. So we thank you, myself and Professor Raman. We thank you all for your attention and
Thank you for giving us the opportunity to be with you to the session. Thank you so much.
So now we are moving to the felicitation. So for the felicitation, I would like to request our chairperson, Professor Madhujita Padia and Rajiv Raman, to felicitate our speaker. So our first speaker, Dr. Medicine, please be on the stage. Now I would like to call Professor Romero. Professor Mehta. Dr. Sharma. And Dr. Bansal. After this, now it's time to felicitate our chairpersons. So for this, I kindly So here is a small announcement. Those speakers who have talked in the next sessions, I kindly request them to give their presentations to us. And you all can move for lunch.